Hello, my name is Frank Oriana, and I will be discussing RT-qPCR, the gold standard for diagnosing COVID-19. So I'll start with COVID-19's relevancy. So COVID-19 was declared a pandemic in March 2020, which has left extensive physical and emotional scars for many. During the pandemic, we saw many governing bodies impose laws that would disrupt normal living. Things such as transportation, food availability, and job security became limited in the hopes of halting the continual viral infectious spread of the disease. Without a doubt, COVID 19 showed the world how underprepared it was when it came to dealing with a global health emergency that put the lives of millions at risk. As of today, there have been about 770 million confirmed COVID 19 cases, including about 6.7 million deaths. With that said, let's learn a little bit more about how it is that COVID 19 works exactly. So, we'll start off with COVID 19 virology. So, COVID 19 is an infectious disease caused by the SARS-CoV-2 virus and is part of the coronavirus family. And like its family members, COVID 19 causes respiratory diseases among other symptoms. The main components of the virus are the spike proteins, the viral envelope, and the RNA genome. That said, the COVID 19 is an RNA virus, and its genetic material is composed of RNA. RNA is a kind of single-stranded nucleic acid that acts as a messenger that tells cells what they need to make and when they need to make it. So, in the case of COVID-19, the viral RNA is the blueprint with instructions of what to produce to ensure survival of the virus. Now, the spike proteins are what give the virus its name corona, which means crown in Latin due to its crown-like appearance. But spike proteins not only give the virus its name and make the virus look cool when you look at it under a microscope or in graphics, as they play a critical role in acting as hooks to attach and crack open host cells to be ready for infection. Now, the viral envelope acts like the armor of the virus, protecting the virus when it's outside of a host cell. And also, the viral envelope is composed of lipid membranes and proteins that allow creation of new viral particles after infecting a cell. So, would you know if you had COVID-19? Well, COVID has a variety of symptoms that one may experience. Someone may experience fevers or cold chills, cough, difficulty breathing, fatigue, body aches, headache, loss of taste, loss of smell, sore throat, congestion, nausea, and even diarrhea. And also, it's important to note that COVID-19 can go asymptomatic. So if you or someone close to you has experienced the mentioned symptoms, you should go get tested. So, and that's really the only way to be sure of that you have COVID-19 is through a diagnosis. But how exactly does that work? Well, that's easy. The gold standard of COVID-19 diagnosis, an RT-qPCR test. So, what is RT-qPCR? RT-qPCR stands for Reverse Transcription Polymerase Chain Reaction. Yes, I know, it's a mouthful. But in simple terms, RT-qPCR is a laboratory technique used to detect and amplify a specific RNA sequence to determine if the sequence is found in the sample. So, in our case, RT-qPCR will determine if the SARS-CoV-2 viral RNA is in our sample that we're submitting. So now you may be wondering, wow, Frank, that sounds cool and all, but how exactly does it detect COVID-19? And all I have to say is I won't be gatekeeping that from you all. So we'll go over that. So the first step would be to collect our testing sample, which comes in the form of a patient's blood, saliva, or mucus. And then after that, we'll perform nucleic acid extraction um, which will include the patient's DNA, which is a double-stranded nucleic acid that is the carrier of our human genetic information. So that's like our template on how we are. And along with DNA, there may be viral RNA in our sample if it is present. So after that, we would then perform reverse transcription, which is when we convert our single-stranded viral RNA into a double-stranded viral DNA. So the reason it's called reverse transcription is because usually DNA is turned into RNA, but in our case, we're reverting or going in the reverse order by making RNA into DNA, as you can see here in the graphic below. So after that, we would have to add chemical reagents needed for the PCR detection, such as our primers and a fluorescent dye. So the primers are used to identify any viral DNA found, and the fluorescent dyes will bind to the ends of the viral DNA sequences identified by the primers, thus serving as a marker for duplication, and I will get back to this. So after we add these chemical reagents needed, we're ready to run the samples for RT-qPCR. So we put them in a PCR thermocycler, which is the name of the machine where you put in the samples. And what happens in the PCR thermocycler is that there will be different temperature cycles, which will trigger the replication of the genetic material inside our samples. And then at the end of our cycle, which can range from 30 minutes to an hour or a little bit more time, depending on the test and who's running it, we're left to interpret our RT-qPCR results. So what does it interpret really? So RT-qPCR determines if a sample has the desired RNA sequences by measuring the fluorescence emitted by the generated copies of viral DNA in the sample after being run through the thermocycler. Okay, so back to what I mentioned with the fluorescent dyes attaching to the end of the viral DNA that was found by the primers. During the time of the thermocycler, since we're going to get genetic replication, if we find the sequence specific to the primer, so in our case it would be the SARS-CoV-2 viral DNA, we would replicate it millions of times over, which would then cause it to emit fluorescence, and the machine will then pick up on whether on how much fluorescence is being detected while it's running, and that will tell us if we have a positive or a negative COVID test. So just to reword that again, in a COVID-19 positive sample, we would expect to generate millions of viral DNA copies that are attached to our fluorescent dye, which is why the more fluorescence detected, the more viral DNA found in the sample, resulting in a COVID positive test. And if we were to be dealing with a negative sample, we wouldn't be generating fluorescence because our primers shouldn't find any viral DNA copies. Thus, the fluorescent dye won't attach to any DNA copies generated while it's in the thermocycler in a negative sample. Thus, no fluorescence or very little fluorescence will equal no viral DNA detection in a sample, resulting in a negative COVID-19 test. So now, just to bring it all back together, COVID-19 is a highly infectious virus that spread rapidly around the world, as it was declared a global pandemic only months after the first recorded outbreak in late 2022. Due to the rate of speed, rapid detection measures became of huge importance because of the attempt to mitigate further spread of the COVID-19 virus. Now, this is when RT-qPCR eventually became the gold standard for COVID-19 diagnosis, as it produced results quickly and efficiently. And with RT-qPCR, it determines if a sample is COVID positive or COVID negative by measuring the fluorescence detected in the sample. And in this case, more fluorescence would equal more viral DNA detected, which would result in a COVID positive test. And no fluorescence would equal no viral DNA detection, which would result in a COVID negative test. So now that we know the how and why RT-qPCR is considered the gold standard of COVID-19 diagnosis, I hope you all learned a lot, and I would like to thank you all for your time, and have a blessed day.